Hello everyone, my name is Sonia. Today we are going to discuss about models of membrane structure. The outermost limit of a cell is composed of living or non-living materials or both. It is called as plasma membrane. It is also called cytoplasmic membrane or cell membrane. Plasma membrane composed of four different types of molecules: phospholipids, cholesterol, proteins, carbohydrates. These are the five major membrane models. Let's see the Robertson model. In 1900, Robertson proposed a biomembrane model which states that biomembranes are made up of lipids. He gave this statement on the basis of observation, transport of soluble substances across the biomembranes. Next model is Gorter and Grendel model. Gorter and Grendel, the founding fathers of our current model of plasma membrane structure, proposed this model in 1925. According to them, the membrane was bilayer, so its surface area should be half that occupied by all its amphipathic lipids spread out in a monolayer. Two tests were carried out independently by them. They measured the surface areas of different mammalian erythrocytes with the help of microscope. They also extracted the lipids from erythrocyte membranes, spread the lipids out at an air-saline interface, and measured the respective monolayer surface areas. They compared these two results and found a cell monolayer surface area ratio was of approximately 1:2 for a number of different mammalian RBCs. Thereby they confirmed their bilayer model of plasma membrane with the polar hydrophilic heads facing outwards, that is towards the aqueous environment on both sides of the membrane, and the nonpolar hydrophobic tails facing inwards, that is away from the aqueous surroundings. This is the diagram of Gorter and Grendel model. This model was unable to describe the membrane function and had false assumptions such as that of plasma membrane consisting of only lipids. Next model is sandwich model or protein-lipid bilayer protein hypothesis. It is also called Davison-Danieli model. This model proposed by Daniel and Davison in 1935, also called trilamina model. They said that plasma membrane is made up of lipids and proteins. Lipids are arranged in bilayer, tails are facing each other and attracted by hydrophobic interactions. The lipids are amphipathic. Proteins are present on both sides of lipids. Glycerol head is hydrophilic and fatty acid tail is hydrophobic. It was the first model that attempted to describe the position of proteins within the lipid bilayer found in membranes. Danieli and Davison proposed a model whereby two layers of protein flanked a central phospholipid bilayer. The model was also described as a lipoprotein sandwich, as the lipid layer was sandwiched between two protein layers. From the speed at which various molecules penetrate the membrane, they predicted the lipid bilayer to be about 6 nanometer in thickness and each of protein layers of about 1 nanometer thickness, giving a total thickness of about 8 nanometer. This is the diagram of sandwich model. Limitations of sandwich model. It assumed all membranes were of a uniform thickness and would have a constant lipid-protein ratio. It assumed all membranes would have symmetrical internal and external surfaces. It did not account for the permeability of certain substance. Next model is Robertson model or unit membrane model. In the late 1950s, electron microscopy provided additional information about the structure of plasma membrane. In 1959, J. David Robertson proposed his famous unit membrane model after studying the cell membranes from electron micrographs of sectioned material. He concluded that the plasma membrane and the membranes of all cell organelles were similar in structure. According to him, the cell membrane is a trilaminar, that is made up of three layers: protein, lipid bilayer, protein. The transparent lipid bilayer is surrounded on either side by dense proteins with a difference in type for outer and inner side. It means there are specific chemical differences between inner and outer side of the membrane. In unit membrane model, the protein layers are asymmetrical and shows structural polarity. On the outer surface, there are conjugated protein and polysaccharides, mucoproteins, while on the inner surface, it is non-mucoid protein. Total external hydrophilic layer is 2.5 nanometer thick. The total breadth of the cell membrane is 6.5 nanometer to 8.5 nanometer. This is the diagram of unit membrane model. Limitations. Although there is no doubt about the similar electron microscopic appearance of nearly all membranes, the strict chemical interpretation of uniformity is no longer supportable. The unit membrane appears as a single trilaminar structure in plasma membrane, site endoplasmic reticulum, Golgi body. Next model is fluid mosaic model, proposed by Singer and Nicholson in 1972. This model exactly explains the structure of plasma membrane. It has two key features: a mosaic of proteins embedded in the membrane and the membrane being a fluid bilayer of lipids, proteins icebergs in the sea of lipids. Lipid bilayer made up of lipids, proteins and linked carbohydrates. Thickness of plasma membrane around 75 to 100 angstrom. Plasma membrane is amphipathic. One layer is formed by van der Waals interactions between two lipid moiety of same layer. Both layers are formed by hydrophobic interactions. According to the model, membrane proteins are in three classes: integral proteins or intrinsic proteins, immersed in bilayer and held in the place by the affinity of parts of the protein for the hydrophobic tails of phospholipids on interior of the layer. Peripheral or extrinsic proteins. It is more hydrophilic. These are non-poly-linked to the polar heads of phospholipids and other hydrophilic parts of the other membrane proteins on the surface of the membrane. Next protein is lipid anchored proteins, essentially hydrophilic, so are also located on the surface of the membrane and are covalently attached to lipid molecules embedded in the layer. This is the diagram of fluid mosaic model. Thank you for listening.